Hello, good morning. Welcome everyone to this IntercropVALUES project webinar, Taking Care of Your Soil, Soil Health and Intercropping. My name is Bogdarka Bojogi. I'm Research and Innovation Officer at IFOAM Organics Europe. We represent the organics sector in Brussels. And it is my pleasure to welcome you today to this webinar. We have with us today Christine Watson, who will give introductions on soil health in the project. She is Professor of Agricultural Systems at Scotland's Rural College. Then we'll hear a presentation on soil health monitoring, from a synthesis of approaches and indicator selection to in-field application using the BioFunk tool method, from Alain Brauman, President of the Association Française d'Études du Sol, and Alexis Dumazeau, agronomist and soil scientist at CIRAD. And then finally, we'll hear from Paul Newell-Price, Head of Soils and Nutrients at ADAS in the UK, on developing practical methods for monitoring soil health on the farm. We will have about 20-30 minutes in the end for a Q&A, so please, if you have any questions during the presentations or after, feel free to put them in the chat function, and then Christine will moderate the discussion and the speakers will respond to your questions. So just a quick background about the project. IntercropVALUES stands for Developing Intercropping for Agri-Food Value Chains and Ecosystem Services Delivery in Europe and Southern countries. We are a 4-year-long Horizon Europe project coordinated by CIRAD in France, and the project is ending in October. So this is the time where a lot of the results and interesting outputs are coming out. So be sure to follow us on the website and social media, as you hear on the slide. And the project mostly focuses on intercropping between cereals and legumes. So the species mixtures that involves the cultivation of multiple species of cereals and legumes in the same field, without necessarily sowing or harvesting them together. This is the definition of intercropping. And while the practice offers numerous advantages, for example, improved resource use, increased yields, or enhanced quality and stability of yields, we know that farmers encounter various challenges adopting intercropping. This can be technical, organizational, or economic. So today we will be hearing more from the soil side of this complex issues. And a little background on the project is that we have 27 participants. Many of those are here today from 15 countries and 3 continents. And we have been producing different practice abstracts, policy briefs, events, scientific publications, and videos on the topic. So if you're interested, there's a lot more to learn about intercropping. And just a reminder that this webinar is a part of a 3-part series in the middle. So we still have one more to go if you're interested about how to process intercrop goods and what happens after harvest and how these cereal legume mixtures will appear on your plate. Then join us next Monday for the final webinar. I will put the information in the chat soon. So without further ado, I will give the word and the control to Christine, who will talk about how soil health is represented in the project itself. Thank you.
Thank you very much, Boggy. Good morning, everyone. I hope you can see my slide. I have the pleasure of coordinating a work package within the IntercropVALUES project, which addresses how intercrops fit within cropping systems, because of course intercrops don't stand alone, but they sit within a crop sequence or a crop rotation. So within the work package, alongside thinking about soil health, we're also monitoring greenhouse gas emissions, particularly nitrous oxide, from intercrops and the following crop. We've also done some analysis from long-term data sets on what it is that an intercrop will bring to a crop rotation. When it comes to the work on soil health in this project, we didn't set out to develop new indicators for soil health. The project is about intercropping rather than soil health per se. So we were keen to think about how we could use assessment tools that were already being utilized in different parts of the world to assess what difference an intercrop was going to make to soil health. I think probably everyone in this webinar knows what soil health is, and the idea that there are three kind of main legs to a three-legged stool, where we have biological, chemical, and physical indicators, and the idea of using those to ensure that soil remains healthy as we move through a crop rotation, but also potentially to pick up any improvements caused by different practices. So from a biological point of view, things like organic matter recycling, soil carbon storage, regulation of pathogens. On the physical side, soil permeability, root colonization, water retention, how workable is the soil? And then perhaps the most traditional of measurements, the chemical ones around soil acidity and nutrient availability. So why are these indicators important? Well, we can use them to give us a baseline for what happens when we change practices and introduce intercropping, as well as assess changes that occur through the rotation, allowing us to look at nutrient use, to look at soil physical properties, and to assess any effects on biology. What we were looking to do within the project was to come up with a simple set of indicators that could be employed across numerous regions. And we needed these indicators to be sensitive enough to detect changes in the system caused by intercropping. And obviously, they needed to be robust and dependent on the science, on known science and principles. So if we think about what the impact of an intercrop onto a following crop might be, we fit that into this idea of different indicators. And we're going to talk a little bit about a soil health scorecard, which was developed in the UK through a big program on soil biology and soil health, which ran for about 5 years, developing these indicators for UK conditions. We're also going to hear about the biofunk tool as well. And within the project, we have tried to combine and to think about the things that we know from the AHDB Soil Health Scorecard and the biofunk tool. So within the project, we have worked on 6 experimental sites. Those all have a replicated experiment where we have sole crop cereals, we have sole crop legumes, and then we have intercrops. And as you can see, they cover quite a wide range of biophysical conditions. So we've got Sweden up in the north, then we've got Scotland, we've got a German site, a Serbian site, a Swiss site, and then a site in Zimbabwe. And, you know, as you'll appreciate, we go from wheat and barley and peas through to maize and cowpea. So, you know, not only do we have different biophysical conditions, but obviously the crops being studied have to fit those conditions. So that's what we've been doing within the project. And to give you an idea of what our soil health scorecard looks like, what we have done is to look at the intercrops to take one set of measurements of our indicators when an intercrop was sown, and to look sort of before and after at soil health indicators. And what we've got here on the left is the first year, and on the right is the second year. So those are 2 different sites. So the starting point is different in each case. And I think what you'll see is here we have a traffic light system where if something is green, that indicates it's a good score. If it's a kind of amber or yellow colour, it indicates a moderate score. And if it's red, it indicates a poor score. So in terms of anything that's amber or green, we would be suggesting that it's continuous monitoring is important to look at how that changes, and anything that's red, we will be suggesting that perhaps interventions are needed. Although with things like macrofauna, which shows up red, and sort of earthworm counts that show up red in the second year, there is also a function of soil type at the site. So what we have been doing is to try and develop ranges for what indicates good, bad, or intermediate in the different sites. And the idea of this is it is farmer friendly. It is things that farmers and advisors mostly can do themselves in terms of looking at infiltration rates and looking at visual evaluation of soil structure, counting earthworms, and then combining that with lab measurements of available NP and K. And I think, perhaps not unexpectedly, we are not seeing enormous differences as a result of putting an intercrop into a system, but we're seeing a maintenance of soil health, or that's where we are so far. So that's our experiment, and I'm now going to move forward to our main speakers. That's just a little bit of background, and we're going to have a joint presentation from Alain Brauman and Alexis Tomaso about the BioFunk tool. So at this point, I'm going to hand the control over to Alain, who I think has already been introduced and will remind you who he is. Over to you, Alain.
Yeah, thank you, Christine, and thank you for this presentation. Everybody hear me very well? I am in Thailand, so I am quite far. And so yes, I am currently the president of the French Soil Science Associations, but I am also a soil scientist and more a soil microbiologist. And from IRD, the Institute of Research for Foreign Countries, so I spend most of my life in Africa and in Thailand. So yes, today we will share the presentation with Alexis Tomaso. And as you see in this title, we will begin by a synthesis of approach and indicator selection. And that will be my part. And Alexis will present more the BioFunk tool methodology. It looks like there is a problem of sharing. I see it looks like some people don't see the sharing of the slides.
No, it should be fine.
Okay, because it's okay.
Yeah.
Okay, that was my presentation. And and the presentation of Alexis, but I think Alexis will be able to do it by itself after me. So this is the title of the presentation already. So yeah, in fact, the presentation I will give here is based on an expertise, a French expertise, which have been done in the last 2 years, which is called Indicasol. And the title of this expertise was Developing Indicators to Protect Soil Quality and Health from a French Perspective. So quickly, what is a French expertise is, in fact, when French ministry or environmental agency like ADEME want to know, have to have a statement, a scientific statement on a given point, they ask generally INRAE, because INRAE is a National Institute for Agronomy in France, to take over this expertise and to take this to lead this expertise with a certain amount of expert. And here you will see that this college of experts was where I belong to. So we were 19 experts with 2 PI. And the main point here that these experts come from different kind of disciplines, not only from soil science, of course, like me, I am a soil ecologist, but some other people coming from more than of humanities, like anthropology, sociology, geography, economy, because soil health is not just a problematic of soil science, it's also a problematic of human science, especially law, perhaps I would say. And so this study have two parts. So it's based, as usual, on an extensive literature review, more than 1,500 peer-review papers, more than 200, 600 review in total. And with sometimes we use great literatures like books, reports. And so totally we have more than 1,800 cited reference. And the second part of the study was devoted to evaluation of these indicators in a given region, French administrative region, which is Burgundy. Perhaps some people know it, especially for the wine. And so we try to do, I will say, an academic work, but after not only an academic work, but to apply this work to a given region. So let's go to soil health, and as usual, perhaps it's good to go directly to the definitions. And so, as you see, the term function is really very important in the definition of soil health. The continued capacity of soil to function as a vital living system. We will see afterwards that the definition is more detailed, but I will focus on function, because function, what is a soil function? And what are they in scientific literature? So this slide is based on 150 papers dealing with soil functions. And so when you analyze all these papers with this on this cloud, so you see that there is a lot of different kind of functions. Could be nutrients, and there is two kind of words here. The first kind of words is word describing object like nutrients, water, soil, carbon, whatever. But there is a second type of words which describe action. Cycling, for example, you can cycle nutrients, you can cycle water, carbon, and soil. You can regulate nutrients, you can regulate water. So in fact, in short, what is the functions? An ecological function can be defined as an action over an object. So you regulate, or you stock, or you produce water, nutrient, carbon. And it's in line with the definition of Sebald et al. In 1998. So within this expertise, we define 6 soil functions and a lot of sub-functions. So first function is regulating water, quantity and quality. The second one is regulating contaminants, degradations, retentions, storing carbon, stock and additional storage, providing nutrients, not only to the vegetables, to the legumes, but to the whole biocenote, which means to the whole soil biodiversity too, which is a little bit different than what you see in the literature. You have the maintenance of soil structure. This was quite complicated because soil structure is not a function by itself. The maintenance of the soil structure can be defined as a function. And of course, in fact, to support biodiversity, you have to support soil organisms, and to support vegetation. So this is the most complicated table of the expertise. So we define 50 indicators. So don't worry, I will go a little bit into more detail. You don't see a lot here. I just want to show you, to explain a little bit the organization of this table. So in red, you see, not in red, excuse me, in green, you see the indicators which have been chosen by Indicasol. And in blue, you see the indicators which have been chosen by the soil monitoring law, which have just been voted 6 months ago, and which are more linked to threat. So our indicator has more linked to functions. And let's go and see it into more detail. I think it will be more clear for you. So here, for example, we have on the side these indicators, and on the other side, you have the 6 functions with the 8 subfunctions. And you see that the link between the indicators in the function could be feeble, could be weak, or could be really important. But in fact, there is quite few indicators which have really a direct link to a given functions. For example, the structural stability have a good interaction with maintenance of the soil structures, or with regulating of water, of the regulation of water quality. Okay, which is not exactly the case here, for example, for particle size distribution or texture, which have a more weak relationship with the soil structure. And on the other side, how these indicators are linked to different threads. You know that there is 8 threads defined by the soil monitoring law, and or perhaps more. And here you can see that, for example, the bulk density we were talking, or the structural, I will take the bulk density here, is linked to a given function, which is the maintenance of soil structure, but it's also linked to 2 main threads, which is topsoil compactions and subsoil compaction. So it's important to know what you want to do with indicators. Do you want to monitor soil straight, or do you want to monitor soil function? And you will not use, for example, the erosion rate will be good for straight, it will not be a good indicators for soil functions. And another problem we have with the monitoring of soil function is that we have around 4,027 indicators in the literature. So we have a lot of indicators. We don't miss indicators. The problems that there is quite few indicators which are directly linked to the monitoring of soil function. That's the problem. This is why Biofunk tool was done in a way. Most of the indicators are linked to soil properties or soil services. And so I will focus more now on the part I really direct in this expertise, which was more the conceptual part linked to what are this notion of soil health and soil quality, how we can define them. And my question was, are these concepts truly distinct? And why I ask this question? Because I think there is a certain level of confusion in not only in the scientific world, but in the farmer world, between these 2 notions of soil quality and soil health. I think it was very important to really distinguish them. And so because, as you know, that semantic clarity is the foundation of any rigorous and scientific reasoning. And so I will see if they share the same definition. I will see, but really shortly, because my time is constrained, a little part of the history of this notion, and I will focus more on the lexical part. So let's go to the definition. As I told you before, soil health was really linked to the capacity of soil to function, but soil quality is the same. We have the capacity of specific kind of soil to function. Yes, there is some difference because in soil health, the soil is more seen as a living system, which is not the case for soil quality. But mostly both of them are linked to the capacity of soil to function and provide ecosystem services. So we have a first problem. These definitions are loose. In fact, they are not really, this definition can't distinguish really the 2 notions. And we see that in the scientific literature, this similar definition leads to a certain confusion of terms. We have at the beginning of the 21st century, the term soil quality really dominates the literature. Mostly that more than 80% of the literature use the term quality. And there was a switch around the year 2010. 2010, you see from 2010, these things come from a paper from Johnson, which is a really good paper. You see a rising up, even an exponential rising up of the term soil health in the scientific literature. And now the term soil health is dominant in the literature, which is really a very quick, domination in less than 10 years. And so this term, we have a certain part of history in this term. Remember, in the year 70, for people like me, which are quite old, we use the term fertility more because we see the soil as a resource linked to one function is the provision of food. But in the year 90s, with the rising up of environmental concerns, the soil was more seen as a capital with the possibility to give a multifunctional, which have a multifunctional roles, forgive me. So not only food production, but carbon sequestration, water quality regulation, water regulation by itself, and so on. And in the year 2010, remember, this was the beginning of the rising up of the term soil health in the literatures. The soil begin to be seen more as an ecosystem which harbor not only physical and chemical term, but a biological life. And you know that the soil harbors the most diverse life in all the different biomes in the terrestrial environment, which means that soil harbors more than 60% of all biodiversity, which is really huge. I will not go further on the term security. So security is quite a new word coming from an Australian team, which sees the soil more as a socio-ecosystem. And so let's go to the lexical analysis, which I think is really interesting here, because when you analyze the papers on soil quality, you can see that most of the papers on soil quality, and you see soil quality is really at the center here of the bubble, of the sphere, is really linked to soil pollution. So, because here, the use of quality is really the same as you use in water or in the air, is a level of pollution of the soil. So you have a target, you have a means, you have database. Here is really clearly defined. Whereas the term soil health is more linked to ecosystem services like carbon sequestration, biodiversity maintenance, which is not, we don't have the same kind of database for that. So we have a more loose term for that. In terms of indicators, soil quality, we use more physical parameters for monitoring soil quality, whereas for soil health, we use more biological parameters, like microbial parameters, but earthworms, macrofauna, mesofauna, like nematodes are also used. And for the agriculture practice, it's less clear. Agriculture practice is really linked to the ecosystem service in terms of soil health, whereas for agricultural practice is more linked to the indicators. So whatever, the conclusion is these two concepts are not similar in terms of history, in terms of lexical analysis, but they share the same definition. So in a way, we have to change the definition and to clarify the concept. And so for us, what we say that the soil quality is more linked to what the soil is, to its potentiality, whereas the soil health is more than what the soil does to its current performance. So soil quality by itself will be linked to the inherent property of the soil. It means if I make a parable that, for example, you can be a tall guy and have the potentiality to become a good basket player. But if you don't train, you will not be a good basket player. So your current potentiality doesn't mean that your current performance will be the same. And so this is what I say, the soil quality is more the potential to fulfill functions, whereas soil health is the actual capacity to fulfill these functions. And so the difference will be very important between this potentiality and this current performance. And so the soil quality is based on defined database at a regional or global. For example, in Europe, we have Lucas, which is a database for that, but it will not monitor soil health. It will monitor the level of degradation of the soil. It will tell you if your soil is saline, if your soil is compact, at a very large scale, which is not at all the scale of soil health. The soil health is much more fine, little, it's local. And that's very important because you will see that we don't need perhaps the same kind of database than we need for soil quality for that. And the time scale is really different. It will be decadal. For example, organic matter will not change on an annual basis. It will change every 10, 15 years. Depends the kind of practice you have. So you don't need to monitor organic matter every year. But for example, this is not the same for soil biodiversity. Soil biodiversity will change on an annual basis. So you see that I hope that it's more clear now what is the difference between soil quality and soil health. And to really clarify that, I will make a graphical representation. So let's think about the same kind of soil, the same. So here you have two soil with exactly the same quality, same depth, same organic matter content, same textural, same climate, okay? But on the first one, you will apply very intense agriculture practice, which end in a high level of reduction of the soil health conditions. So the soil health represents 50% of the potentiality of your system here. Whereas if you use more sustainable practice, you will not degrade your soil at the same rate. And so you are more close to the potentiality here. The level of soil health is 80% of the potentiality. So in the I will say ideal world, it will be nice to be able to measure the difference between potentiality and performance. But this is possible for only a few indicators, like for example, water reservoir, things like that. But it's not always possible for all indicators, especially biodiversity indicators. But what you can do is you can, if you are in the same pedological context, can just make a relative comparison between soil A and soil B. And this is exactly what Biofunc tool does. And so this is the end of my presentation. I hope I don't really explode the time. And so if you want to learn more about these studies, there is a report which is in English too, not only in French. We have done a book synthesis which will be in English soon. But, and so you have all here the link to load this report if you are interested. There is for people who don't want to read 700 page, you have an extract of 12 page. Thank you for your attention, and I will be pleased to answer to all your questions.
Thank you very much indeed, Alain. That was fantastic, and a great overview of some of those thorny issues, and those language issues we have all struggled with. So thank you very much. We're going to keep questions until the end, but I would encourage people, please put your questions in the chat, because then we can ensure that we get them at the end. So thank you, Alain. I'm going to hand over to Alexi to tell us, I think, more about actually using the Biofunc tool in practice. So the floor is yours, Alexi.
Thank you, Christine, for the introduction. So I will indeed move a bit more practically on the Biofunc tool method. So as Christine said, under the IntercropVALUES, we combine actually several indicators from these methods. And today I will present you the concept of the Biofunc tool and also the indicators and few examples of and case studies of the use of this method. Please confirm me that you see the next slide is good. It's okay?
Yes.
Yes.
Okay, perfect. So just let me start with the small illustration showing the basis of the development of Biofunk tool, and we actually developed it with Alain and other colleagues, and we found that the current assessment of soil health was actually made from summing up several properties of soil. And in this case, we illustrated with this dog. We found out that the more the better. It means that the higher physical, the more chemicals you have in the soil, the more the higher biological properties you would have in the soil, the higher your soil health would be. But actually, we asked ourselves if this way of assessing soil health was the good metrics. And to do so, we had mainly examples of the tropical areas, seeing that many soils in the tropics are very low in terms of stocks, in terms of content in the soil, whereas they are functioning very well. This is especially the case of this savanna in Ivory Coast, where you have a very huge biomass produced by soils. But when you measure the stocks or when you measure the properties of those soils, they would be very low carbon, very low nitrogen, very low phosphorus. So we were asking, are the current ways of assessing soil health the good metrics to understand the processes and to understand how the soil system is functioning. And so from a study of Kibble-White, we proposed to change a bit the metrics and the way how we understand and measure soil health from the, like I would say, classical way of measuring soil properties, for example, soil organic carbon, nitrogen, total nitrogen, earthworm abundance, etc., to a more functional way. So what we call the integrative approach, not focusing on the stocks or the contents in the soil, but rather focusing on the processes in the soil and the result of the processes in the soil. And actually, most of those processes in the soil are mainly led or driven by soil biological components. So we aimed at gathering methods within this Biofunk tool that would allow us to understand the results of the activities or of the interaction between soil organisms, rather than the dividing the components of the soil system. So to build this Biofunk tool, we targeted 3 soil functions, and those soil functions are a result of the soil biological assemblage activity. So we focused on soil carbon transformation, nutrient cycling, and soil structure maintenance. We discarded the pest regulation because it was very linked to cropping systems, and we wanted to be able to use this method in all cropping systems. And so to select indicators, we are always asking us this question before implementing any method to assess soil health. We are like, which indicator should we select to be able to detect the change in terms of management practices? And so we had 3 main criteria to select the indicators. The first one was to have some measurements, some methods that allow us to assess the result of soil biological activities, the functioning of the soil. So we focused on 3 different soil functions that I already detailed you before. And we discarded all the methods that are focusing on pure description of the soil composition or contents. So all the component, the physical, chemical content, or even the abundance of organisms were discarded. Our objective were to measure how those interact with each other and what is the result of those different activities or interaction. The second criteria was to keep some indicators that can be implemented in the field, because we wanted to tackle the reality of the soil and not the potential. And the third criteria that we we defined was to have some indicator that can be easily implemented. So with a low-tech characteristic for time and cost effectiveness, to be able to replicate it and to be able to better understand also the soil spatial heterogeneity. And finally, through these indicator selections, we screened all the literature, and we finally selected 9 indicators that met our expectations, which are gathered in the BioFunk tool set of indicators. So really keep in mind that all those indicators are indicating the connections between soil organisms, soil biological compartment within the physico-chemical context of the soil. So these are the 9 indicators we selected. So the first one is the permanganate oxidizable carbon that assess the available and labile carbon in the soil that is easily available for soil organisms. The second indicator is an assessment of basal soil respiration. And the third indicator is an assessment of the fauna activity through the lamina-based method. For the nutrient cycling method, we focused with 2 indicators. The first one is a classical nitrogen extraction, available nitrogen extraction from soil. And the second one is working with ion exchange membrane that are membranes that we bury in the soil for a while. And those membranes mimics the plant rooting systems and thus give us some ideas about the availability of nutrients in the soil. And then for the structure maintenance, we have 4 indicators linked to the soil infiltration, soil aggregate stability, and visual evaluation of soil structure. So you can see that actually among those 9 indicators, some of them were presented by Christine in the beginning. They were included in the IntercropVALUES study sites, and others were discarded for several reasons, such as not that easy to implement or other reasons. So I will not go into detail too much on the method because the time is running, but just for you to know that all those methods already existed in the literature. Actually, we didn't invent any methods. We just combined them through this functional approach and then gathered them. So I just pass it very quickly, but if you have any questions, don't hesitate. We can discuss about those methods later. And so the principle of this BioFunk tool is to measure in the field those 9 indicators. We mainly focus at the topsoil because this is the soil layer where we have the highest biological activity. And then we aggregate the results of those those 9 indicators within those 3 soil functions through a scoring. And this scoring is made from statistical analysis, and it's between 0 and 1, and the higher the score is, the better the soil health is. And what is very important to know is that those indicators are most of the time used to compare practices within a given pedoclimate. So we are working on soil health and trying to understand the impact of management practices. So we most of the time work without any bias from the soil type or the climatic condition. Or we also use this method to follow up the temporal transition of management practices, repeating the measurements with time in the same plot, for example, following intercropping or other management practices. So I will briefly explain you, share with you some examples. I will go fast because the time is running fast. Actually, most of those examples are already published, so don't hesitate if you have easily access to scientific literature to read it. Otherwise, we can also send you some documents that explain the result a bit more easily, I would say. So the 3 examples that I will share are the first one in Thailand on the interrow management of rubber plantations, the second one on conservation agriculture in Cambodia and New Caledonia, and the third one on alley cropping in France. As I see the time is running, I think I will skip the first one because it's also very typical from Thailand. So I will skip the first one and directly go to conservation agriculture and alley cropping. Okay, so let's go to conservation agriculture. So this experiment was implemented in Cambodia, and it was a long-term experiment. And we had a different cropping system with various tillage and also biomass restitution. That was actually the key parameters of those conservation agriculture. So the first one, first treatment, we had tillage and low biomass restitution. And then we had 3 systems under conservation agriculture with no tillage and higher biomass restitution, with soybean and soybean maize rotation. And the result, so this is the kind of result we get from BioFunkTool. You see our score between 0 and 1. And you can see the 3 colors of the 3 soil functions. So it's an aggregation of all the measurements that we did in the field. And you can very clearly see that for this system, the method was very sensitive to the management change, and it indicated higher values for the 3 conservation agriculture management practices compared to the conventional tillage system. We implemented the method in a completely different context in New Caledonia with completely different type of soil. And we wanted to check the robustness of the method. And we observed that in this New Caledonian context, we also observed our 3 functions positively affected by the change from conventional tillage to conservation agriculture. Let me switch to the example of alley cropping in France. So in this case, we worked with 3 different tree species, maple, ash, and pear. And we wanted to know if there is spatial heterogeneity within alley cropping systems. So alley cropping systems are agroforestry systems where you have tree lines and then annual crops cropped in the middle of the tree line. And for this example, we had we measured 3 points, 1 point in the middle of the tree line, 1 point in the beginning of the cropping alley, and 1 point in the middle of the cropping alley, C2. And our objective were to know if, especially the C1 in the beginning of the cropping alley, was more close to the tree line or was more close to the middle of the cropping alley. So these are the results. So TR is a tree line, C1 is the beginning of the crop alley, and C2 is the middle of the crop alley. And you can very clearly see that C1, the beginning of the crop alley, even though this point received the litter from the tree, received also the roots, had the root systems of the tree very close by, we can see that actually it reacts exactly as the C1. So all the cropping alley was actually homogeneous in terms of soil health and was not sensitive at all from especially the litter fall from the tree of the tree row. And we could observe that our 3 soil functions were very sensitively affected. Structure maintenance, nutrient cycling, and carbon transformation was much lower in the crop alley for both C1 and C2 compared to the tree row. So this leads me to the conclusion. So BioFunkTool gathers functional indicators linked to the soil biota and linked to the soil biology. What is very interesting to our point of view with this method is that it can easily be linked to agronomical management and management practices. Especially for the example of conservation agriculture, we observe that actually conservation agriculture improves the soil health in various contexts. So the 3 soil functions were positively affected. And for the alley cropping systems, we observed that the soil health was improved with the tree at the plot scale, and carbon transformation and soil structure maintenance were especially affected in the cropping alley. We observed that C1 and C2 were much reduced in the cropping alley. And we can easily link it with the management in the cropping alley that was very intensive, actually. There were high tillage and high, it was actually managed as a conventional system. So we could think about less intensive management system in the cropping alley. And in this case, maybe we could get higher benefits from the tree row in this kind of systems. This is only for sure linked to soil. Then we need to understand other trade-offs linked to yield, socioeconomic factors, and others to be able to understand the cropping system as a whole and not only soil oriented. Thank you so much for your attention, and very happy to answer the questions later. And also you can find a lot of documentation online on our website and with the scientific publication.
Thank you very much indeed. That was fantastic. Great to see those examples from different parts of the world. Given the time, we're going to move swiftly on. And I'm going to invite my UK colleague, Paul, to join us. If you can, yeah, there you are, Paul. Lovely to see you. I'll hand over straight over to you.
Thank you very much, Christine. So if I think I've taken control. Is that moving forward?
We see a yes. Perfect.
Yeah, great. Okay, I'll work on that. Great. No, thanks very much. Yeah, Paul Neal-Price. I'm a soil scientist from ADAS in the United Kingdom. I head up our soil and nutrients team. And today, yes, I'm going to be talking about developing practical methods for monitoring soil health on farm. Christine has introduced, given us an overview of the soil health scorecard, which was developed for our levy board, the Agricultural and Horticultural Development Board in the UK. So I'll be having a look at that with respect to soil health. So in terms of what I want to cover today, a few more definitions of soil health. I won't go into detail because we covered that a fair amount already, but a little bit about assessing soil health, but particularly the soil health scorecard approach, and a little bit more detail on that in terms of the different indicators and interactions between the different indicators. And then looking about improving soil health, the role and benefit of soil organic matter in that, and then the principles of regenerative agriculture. So talking about sustainable soil management and intercropping's potential place within it. And maybe we can talk a little bit later on about what sorts of metrics, measurements we can use to assess the impact of intercropping on soil health. So first of all, yeah, just some quick definitions. We've heard a few already. So we've got various definitions of soil health. There are multiple out there, but one of them is the ability of the soil to act as a living system to sustain in the long term its most important functions. We've always thought about soil health as being similar to soil quality, but adding the living system to it really. And as Alan said, the emphasis on sort of function and doing stuff rather than just being in terms of quality. And then in terms of other definitions, we've had a definition for soil quality as fitness for use, and soil sustainability is really around the ability of the soil to function in the long term, so to continue to do what you want it to do, and that's about delivering a range of ecosystem services, which you can see on the right there. I won't go into the detail, but we're wanting our soil to perform a lot of functions and deliver a lot of services. So yeah, just thinking about the key aspects of soil health, we like to think about it in terms of what the soil does inherently, but also what it does in terms of changing properties. How you manage it, how you manage the soil will change those dynamic properties. And the crop will also have an effect in terms of the residues it leaves behind, feeding the microbial community, getting roots into the soil, which have an effect in the rhizosphere and development of what we call rhizosheaths to create stable aggregates within the soil, which perform their own functions. So we have these interactions between what the soil inherently is in terms of its texture, its depth, its drainage properties, its stoniness, those dynamic properties which can respond quite quickly through management, but also respond slowly over time. And then what's happening in terms of the crops that are being grown and the sorts of crop rotations that you've got in place. And intercropping would be a part of that. Moving on then, in terms of assessing and managing soil health, how do we know if the soil is healthy? What do we need to measure? We've talked about that a fair bit already. How do we benchmark, interpret these measures, and how can we improve soil health? So we can do a fair bit just by looking at it, to look at that soil structure and seeing the shape and size of soil aggregates and the continuity of pores and the size of pores within the soil. But we know we do have, as Christine mentioned earlier, we have chemical, physical and biological properties. They are interacting, and we know if we change one, it changes the other. For example, with the sort of physical, think about physical properties. If you live in a house or a flat, it has a physical structure. You have rooms, you have staircases to get up and about. If you bring a bulldozer into that situation and take out the staircase and the supporting walls, all of a sudden you find it's a bit more difficult to move around your living space. It's a bit like that in the soil. If you come around, you come in and disrupt it, you disrupt the physical structure, it becomes a bit harder for certain parts of the biological community to function, to do their function. So you might be changing that biological community. You also change the chemistry because you're changing the environment in terms of the availability of oxygen and the sorts of microbial community that will dominate. So it's a real interacting system that we're talking about. So no one indicator will cover all aspects of soil health. We have to pick a number to try and summarize what's happening, get a picture of what's happening in terms of the performance of the overall system. So with that in mind, through the Soil Health and Soil Biology project, Soil Biology Soil Health project for HDB, Agriculture, Horticulture and Development Board in the UK, there was a team involved in developing the soil health scorecard approach, which is a health check for soils. And we've got our chemical, physical, biological indicators, which form part of that. And as Christine showed earlier, we have this traffic light approach, where a red means there's something not quite right, go and investigate. The orange means that you should be monitoring the situation. And then the green means just continue to monitor things should be relatively okay. So in terms of the physical, chemical and the biological, you can see that there were a number of candidate indicators for each, but we eventually focused down on VES, the visual evaluation of soil structure, for the physical, along with penetration resistance and bulk density. For the chemical, we've got pH, which is a driver of a lot of what goes on in soils. We've got routine nutrients, which is the pH, the phosphate, phosphorus, potassium, magnesium. And then we've got soil organic matters being a sort of key driver of what's going on. And then in terms of the biological indicators, we've got our ecosystem engineers, those earthworms, which move the soil around and create a lot of pore space and aggregates. And then we also thought it would be good to integrate some sort of microbial measurements, so respiration, a measure of the activity of the microbial community, and then microbial biomass, a measure of its size. So some of these are still developing areas in terms of our understanding in terms of how a measure of respiration actually links to function and soil health. But we have a lot of measurements. We know what a high, medium and low value is, but linking it to actual function is still a bit of a challenge. So just to go into some of these in a bit more detail for those that aren't familiar with them. So the visual evaluation of soil structure was developed by a team from SIUC and Aarhus University in Denmark. So a team including Bruce Ball, Lars Monkholm and Tom Beatty and others got together to develop this. And it's a measurement of the physical structure of the soil in terms of the size of aggregates and the porosity of the soil and the shape of those aggregates as they come out of the soil to give us an idea of how compacted the soil is. And you see, you've got some examples on the right there in terms of a sort of fine crumb structure which performs well and drains well compared to a sort of platy, large sort of angular structures where there's more of an issue in terms of drainage and aeration. And there we've got the sort of scores and the traffic light system. Next one is earthworms. So some guidance there in terms of how to do it. So autumn or spring being the best times to assess the earthworms, and linking it to the soil structural assessment. 3 to 5 pits per soil management zone, and then counting the worms in a 20 by 20 by 20 block, which is not 20 centimeters cubed, that's 8,000 centimeters cubed. But how many earthworms can you find in 5 minutes? And you can sort those worms into adults and juveniles, and also into ecotypes. And then the scorecard then categorizes the numbers that you find into the traffic light system in terms of, you know, whether to investigate further, or whether the situation looks reasonably okay. The numbers are based on the visual soil assessment guidance, which is originally from New Zealand. So the numbers, the ranges will be revised over time as we gather more data and get a better understanding of what a good number is in terms of earthworms and the different numbers of the different ecotypes. Moving on to soil organic matter levels, what is a good level of soil organic matter? The approach used here was looking at how organic matter varies with climate, but also clay content and land use. So we've split the categories in terms of the land used and the rainfall region. So here we've just got some ranges by clay content for arable in a dry part of the country, and the benchmark system just compares what you've got in your soil with these ranges to determine, where you are within the population, typically whether you're at sort of high or low value. So we know soil organic matter is very important. Having soil organic matter and having regular additions of soil organic matter is very important to fuel the soil, provide energy for the organic community, the organisms within the soil, and it has all sorts of benefits. So we know it's good for structure. It improves soil structure, water holding and infiltration. It improves biological activity, but we know some organic amendments are better than others in terms of improving that biological activity. It improves rooting, so it's good for cropping within agricultural systems, reduces runoff and erosion and waterlogging, and generally improves the productivity and the resilience of the agricultural system. So when you get a perturbation of any sort, whether that's a waterlogging incident or a compaction or a chemical perturbation, soils with higher organic matter tend to bounce back better than soils with a lower organic matter content. So you have a more resilient system, and you've got lower costs overall because it's easier to work the soil in general. So lots of reasons for maintaining organic matter content in soils and increasing it where it might be low. So yeah, this slide just gives it's another example of a soil health scorecard, showing across the top 3 zones, 1, 2, 3, and the traffic light system in terms of investigating, monitoring, or no action needed. And we see that generally we're in oranges and greens for most of the indicators there, but the key issues are with sort of quite high VES scores of 4 for two of the zones there, indicating a limiting layer within the soil, so some clear signs of compaction, and also earthworms are low for that soil, so indication that maybe the earthworms, for whatever reason, don't have as high numbers as you'd like. Maybe there's been frequent cultivation, maybe that there aren't a lot of residues being added to the soil to provide food for those worms. So it just gives you a very quick overview of what's going on for that soil. So what can we do to maintain, increase soil organic matter? We talk a lot about sustainable soil management practices, and then there's been a lot of talk recently about regenerative practices. The two are quite similar. So this is quite big picture stuff, but in terms of making large differences to soil health, we can introduce grass to the rotation. We can apply organic materials. We've done a lot of research showing how quickly adding organic materials can improve soil physical, biological, and chemical properties. We can use cover crops or green manures and incorporate residues within the rotation. That can help. We can adopt or reduced or zero tillage, so less disruption to the soil system. And we can introduce practices to reduce soil erosion, such as cover crops, keeping the soil covered. So, yeah, the regenerative agriculture is really a means of improving soil health, recovering soil health through sustainable soil management practices. Just very quickly on cover crops. There's quite a lot of work being done on cover crops, on the various benefits that they provide, but also the characteristics of their rootings, either as monocrops or in mixtures. So some of that work that's looked at how the rhizosheath, so the soil aggregates around the roots, varies between monocropped cover crops and mixtures of cover crops. Reads across fairly well to how intercropping may be improving soil health. And the next slide is just a bit of an aside showing how use of cover crops can really help in terms of reducing nitrate leaching and improving nutrient cycling through the rotation, retaining nitrogen in the cover crop and supplying it to the following crop. Another good way to improve soil health is introducing livestock within arable rotation and introducing temporary lays. Again, that can have multiple benefits. One of it, which is improving soil organic matter, and then also there would be follow-on effects to soil health overall. And the next slide shows that in quite big picture. This is data from Rothamsted Research, kindly provided by Dr. David Paulson. And it's showing on the left-hand side that you've got the Highfield site at Rothamsted Research, which was originally in grass, and you can see there at the top line is showing what's happened in terms of changes in soil organic matter over time. When the field is kept in grass, you get a continued slight increase in organic matter, maintaining soil health, you would hope. And then below that, you can see what happens to soil organic matter when it's converted to arable or lay arable rotation. So you have a reduction in organic matter when it's down to an arable rotation. Also a reduction within an arable lay, but slightly less rapid reduction. And then on the right-hand side, we've got land use going the other way. So it's starting in arable and then going to either grass, arable lay, or arable. You can see the top line showing where it goes from arable to grass, so how quickly over a few decades the organic matter increases. In the arable situation, it's staying the same, maybe reducing slightly, and in the arable lay, it's increasing slowly compared to the arable to grass. So it just shows that quite large changes are needed to quite large changes that are needed in management or land use to see a change in soil organic matter. So the key message is really from the work we've done is that assessment of soil health requires this integrated approach, so linking chemistry, physics and biology, and then linking that to function and ecosystem service delivery, if you can do that. Evaluating the impact of management practices is very important, tracking changes over time. The soil health scorecard is a very useful way of doing that. But you can't expect to see very rapid changes. number of ways to improve soil health, largely linked to improving soil organic matter, and just emphasising the importance of good soil structure, because without that, then you have a number of challenges in terms of how you manage your soil. And I'll end there.
Thank you very much indeed, Paul. That was a great run around some of the sort of practical indicators. Can I ask Boggy to stop sharing the slides and ask Paul and Alexi to put, well, Paul's got his camera on. Alexi, could you put your camera on? Brilliant. And we can have some conversation. I don't see any questions in the chat, so you're going to have to put up with my questions instead. So I guess the question I would like to ask both of you is, what are the indicators that you would expect or you think might change as a result of intercropping? I don't know who wants to have the first shot at that.
Yeah, I mean, from what I've seen that there's been a bit of work, as I mentioned, on the rhizo sheath, so stability of aggregates around the roots, and there seems to be a relationship between root length density, and the number and length of root hairs, and the stability of this rhizo sheath, and the number of aggregates around the roots, and it seems to be species related.
Yeah.
And if you can get the right mix of species in your intercrop, then that should have an impact on aggregate stability, you would think, but it's still an emerging area. So yeah, soil structure and aggregate stability is one, because just by getting roots in the soil for longer, and maybe a greater variety of roots, there's potential for a greater variety of function potentially.
Yeah, and I think you're right, and I think that applies across. I mean, a lot of the IntercropVALUES project is about cereal, grain, legume type intercrops for food production, but I think intercrops are much wider than that. You know, when we think of sort of grassland mixtures and stuff, and in fact agroforestry, as we heard about. Can I pass over to you, Alexi? Do you have some thoughts on that?
Yes, I think I would say that it's always a question that people want, which indicator is the best, which one. And to my point of view, what I observed is that sometimes it's really unpredictable. And that's also the importance of implementing multi-criteria measurements, like not only focusing on one like soil organic carbon or two indicators. Sometimes you just thought that it wouldn't change with these management practices, and you have very unexpected results. So I would say that it's very important, to my point of view, to implement several measurements at several parameters together to be able to hope to observe some changes. And then I think that there is it's always also a matter of contrast. It means that I think you have, for example, the biofunctional indicators that are infielding indicators. For sure, you will not be able to tackle some very minor change like the rhizosphere or something like this that are very mechanistic and very small, that you would get with some more advanced method, I would say. But then the contrast of, I guess, if you implement intercropping for a long time, or if you have like a change in terms of density, or a change in terms of I don't know, tillage or management of the harvesting or something like this, that would help to find differences. So I think the question would not only be on which indicator answer to what, because I think we'll never solve this question, but contextualizing how this question would be much more important. And also really the key point is sometimes we really observe unexpected results, and it's very important to enlarge the set of measurements to be able to hope to get something.
Yes, thank you very much for that. I mean, I guess one of the things that we were focused on within the project, as you know, is kind of farmer friendly and advisor friendly type measurements, and I think it's something that Paul and I have talked about in the context of a recent UK project about what are the indicators that farmers want to see? And I think one of the really challenging ones is soil biology. I mean, yes, earthworms are nice and easy. Hardly an iconic puddly species, but it is relatively easy to get farmers to count earthworms. But I wonder how close are we getting to other really simple indicators of biology in the field? Any thoughts from either of you on that?
Yeah, as you say, the joy of earthworms is its simplicity. You can take a spade out in the field, you dig out a small block of soil, you count them for 5 minutes, you've got a result. And it's just the nature of them. They're easy to spot. You can identify them to ecotype level fairly easily with limited training. To take it further than that, if you're going down the sort of mesofauna route to springtails, mites, nematodes, then the sort of level of expertise that you need and the time that you need it's on another scale, really. So it is difficult. But I agree with Alexis on trying to build up this picture with a number of different indicators. So the other indicators are also that they also help you understand or give you clues as to whether you have met your potential in terms of your soil health. Just things like soil color can be an indicator. If you compare the soil in the middle of the field with at the edge of the field, for example, and if it's a lot lighter in the middle of the field, then it's an indication that you might have lost a fair bit of organic matter. But at the same time, you need to be fair to the agricultural system, because you can't expect it to perform in the same way as a hedgerow would or a woodland would. So, but yeah, just building up that overall picture of how the soil's behaving, and if you know the physical structure is poor, you know automatically that will be challenging for certain organisms within the biological community in the soil.
Thank you, Paul. Alexi, can I pass to you?
Yeah. I definitely agree with Paul as well. I think the key point on biology is that the small taxa, actually, they are very difficult to assess, to identify by the farmer. You need specific skills for that. And actually, it's a bit the bet that we did with Biofunk. Like Alain, he left us, but he would say that actually he's a microbiologist initially, and he was a bit like disappointed by all those methods that you just sample only one sample per plot because it's too expensive, and then you need a lot of expertise. And we know that soil is very heterogeneous, and we can't only take one or two samples to understand the diversity. And especially because we were working in developing countries, and then you don't have the machine to do it properly or something like that. So actually, instead of looking at the biology as the diversity per se or the abundance of organisms, we completely switch the game, playing with the activities of those organisms. And actually, you have quite a bunch of methods now that exist on this, such as the tea bags. Maybe you've heard about that. The lamina baits that we use as well. You have in France also, some farmers did the underwear. They buried the underwear under soil. I don't know if you heard about that. So these are actually linked to the biology because it's soil activity directly. You don't know who is active or not, but at least you know your soil is active. And I think these kind of functional measurements, we are lacking of it because we don't have that many available. But this could be easy and very informative to know the biological state of the soil. So I think maybe we should rather switch on the functional methods and trying to develop more, because we are lacking of it for sure. But working on this instead of still working on the pure diversity or abundance of organisms, because we will lack of methods, especially if we want to tackle the farmers and work and find some indicator that can be directly used by the farmers.
Thank you. Yes, Simon, I completely agree with you that I think the idea of, there's been a lot in the UK on soiling your undies and burying pants, and you know, the idea that I think was developed a long time ago at Cornell of just putting in a square of cotton into the soil. And you know, we've certainly tried. We have a long-term experiment here with soil at different pHs, and you get a very different answer. So yeah, I think and the tea bag index as well is one that perhaps people on the call might like to look up as well. We do have a couple of questions in the chat, which is great. Please add more. We still have a few minutes. So the first one is from Sophia in Portugal. I'm curious about how the Biofunk tool works in the field. Is it an equipment? Is it a kit? How does the sampling work? And then perhaps I'll give you a second question as well on the tool, asking if the Biofunk tool is designed to evolve over time. Are you updating indicators, and have you got new ones? So perhaps I can hand that one to you, Alexi.
Yes. Thank you for the questions. So the first one, how does it work in the field? Is there an equipment? So actually, in the beginning, we developed this method based on a method that existed in the literature initially. So we just combined some method that existed, but we knew that many people wanted to apply it without like having to think about like the supplier or everything. So recently we got we have a French Fab Lab, is it? I think like a French small group, small company that make with a 3D printer that make all the equipment, and now you can easily get the equipment. So I can give the website in the chat or something like this if you wanna get the equipment. The only one thing, but one small webinar like this would not be enough, is that there are a lot of precaution before using the things. Like you need to be careful on the temporal dynamic, you need to be careful on how to implement the tool. So it's we can discuss more, Sophia, I think, if you need some specific, if you have some specific questions on the method, because it's always very interesting to have 9 indicators, but then it's 9 measurements that you need to understand, that you need to implement. So it's also a bit, it can be a bit complicated. So I think, yes, I can share the website to buy the material. This is now available, but actually everyone can do it, because it's all methods that exist in the literature before, and that are conventional methods. We didn't develop a method by itself. And then designed to evolve over time. Yes. So we are also, because actually the method was developed maybe 10 years ago, something like this. And we are working on trying to make it more easy to implement, working with new technologies such as the Arduino measurements to reduce the cost as well. So we have a team in Montpellier in France who are working, one colleague at least, who is working on that, trying to optimize the set to be able to make it even more easy to use in the field. But to be honest, it's not a kit. So you need to, it's still a lot of materials, still a lot of methods, and you can buy the material one by one, and it's almost a kit, but it's not like a small box that you would use in the field, like a field test that we can see sometime. But if you have any, I will send you the website of the thing. And if you have any other questions, don't hesitate to contact us by email, and we would be happy to answer.
Thank you very much. I think if there aren't any more questions, we can look towards winding up the webinar. I just wanted to ask to give each of the two of you the chance to give us a last thought around intercropping, because we're here to discuss the IntercropVALUES project. Do you think that we can encourage farmers to take up intercropping for reasons which include looking after their soil? Is it a reasonable thing to be encouraging people to use this as a way of managing soils?
Yeah, I think in the mix, the evidence that intercropping directly benefits soil health is limited currently, but it's logical that if you have a greater diversity of plants within your rotation, then that should improve soil health, because you have differing rooting characteristics as I say, it could have an effect on the rhizosheath. So, and we know there are relationships between plant diversity, plant types, and microbial community. So, if we think of soil health as being a living, it's the living system element of it, then affecting that microbial community is important. Whether to what extent it has a practical impact in terms of helping your farming system, it's less clear. As I said in my presentation, the big hitters are more like changing the diversity of the rotation as a whole, but intercropping could come into that, integrating livestock, adding up, making sure you are recycling organic matter within the system with organic amendments or crop residues. So those are the sort of bigger impact methods, but I think intercropping could be a very important part of that, and there's lots of other reasons for using intercropping.
No, thanks, Paul.
Nutrient dynamics,
that's a really nice. I think that sort of message about diversity, I think, is a really nice sort of takeaway one. I don't know if you have any further comment, Alexei. Just a last comment.
Just to say, but I think it's not really linked to intercropping, because honestly I don't have a lot of experience on the temperate intercropping. But anyway, I think what the best to convince the farmer and everything is to try to find, as we do in IntercropVALUES, some small indicators, quite easy to understand, quite sensitive, and especially to combine it with other parameters, because the farmer will not focus only on their soil. So we need also to be able to give some information on the yields, on the economic barrier, on the social barrier, and to gather them together, because otherwise focusing on soil only will not be enough, and will not be enough to change. But we need to put soil in the loop as well, for sure.
Of course, we always need soil in the loop. I think that's a great place to end. And just before I hand back to Boggy, I just want to say thank you very much to all three of our presenters for fantastic, interesting presentations. There are many people in the chat asking for copies of slides, and I know Boggy will talk about that. So I just want to say thank you very much to the speakers. I also want to say thank you to my colleague Paul Hargreaves, who set this up today and invited the speakers and did all the hard work. Unfortunately couldn't attend, so I'm just the substitute that's been sent in to do the job here, but that's no problem. So thank you very much to Paul, and also thank you to the team, to Boggy, but also to John and Sonia for setting this up and doing all the publicity around it. So with that, I'll just hand over to you, Boggy.
Thank you very much, Christine. I echo all the thank yous to the team, and you did an excellent job moderating the discussion and stepping in for Paul. So thank you for that as well, and to all of our presenters. It was a really nice set of presentations. Whether you're a novice to soil health or more experienced, you could take a lot out of it for sure. And be sure to also join us for the last webinar of the series about processing on Monday. All of them will be available on the YouTube channel of the IntercropVALUES project. So that's intercropvalues.eu. And we will confirm that the presentation should also be uploaded with the video, so you will be able to reach it afterwards. So with that, Sonia, if you don't want to say anything more, then thank you very much also to Iniciativas Innovadoras, and we'll see you next time. Thank you.
Thank you, and bye-bye.
Thank you.
Thank you, bye.
Bye.